but as we discussed, probably a good time to go ahead and get started. It's 3 minutes after, and the recording has started. So it's a real pleasure to welcome all of you to the first of the High Fidelity seminar in this year's seminar series from the Frederick National Laboratory. And I'm in particular really delighted to introduce our seminar speaker today, Sam Michael from the National Center for Advancing Translational Sciences, or NCATS. And Sam currently serves as the Director of Automation and Compound Management, as well as Chief of the Information Technology Resources Branch. And Sam joined NCATS in 2005, and this was at the beginning of the NIH Molecular Libraries and Imaging Program, which was a large common fund initiative that was intended to make small molecule science and high throughput screening accessible to the public sector. And NCATS was the first screening center in the Molecular Libraries Probe Production Center network, which included 11 sites located throughout the United States. As the Director of Automation and Compound Management, Sam provided outstanding leadership and established a world-class chemical compound screening facility at NCATS. And Sam and his team created, operated, and maintained a suite of automated high-throughput robotic screening systems, which enabled the development of hundreds of chemical probes that investigators worldwide use to validate new drug targets and chemical leads for the development of new drugs for dozens of currently untreatable diseases. So Sam was recognized for his leadership and substantial contributions in 2012 when he was named the NIH's Federal Engineer of the Year. And this was an award that was sponsored by the National Society of Professional Engineers. In working closely with Sam over nearly 10 years, I've seen how he's constantly thinking years ahead to keep NCATS at the bleeding edge of technologies and innovation. And this forward thinking is what led Sam to take on the role as Chief of the Information Technology Resources Branch, when he realized that the major future challenges NCATS would be facing would be computational and information technology. Notably, Sam and his colleagues recently earned the U.S. Department of Health and Human Services, or HHS, Secretary's Award for Distinguished Service for the NCATS National COVID Cohort Collaborative Data Enclave, as well as the HHS Award for Meritorious Service for the Open Data Portal team's contributions to data sharing and drug repurposing to accelerate discovery. So today, Sam will tell us about a specialized platform for innovative research exploration. Please welcome Sam. Sam, I'm going to turn the wheel over to you.
Nate, thanks so much, and really a wonderful introduction. And just for the record, for the folks that I classify myself as a plumber. Much of what we do, I would say, is a support function, and it's connecting pipes and making sure that things run smoothly from an operational perspective. I am going to talk about Aspire today, and I just want to confirm everyone can see the slide, the screen I'm sharing.
Yep.
Thank you, Nate. But really, this conversation is about scientific collaboration networks. And I mentioned earlier, I see Doug Auld joined, and I think it's fitting because Doug was, someone that worked at NCGC at the time with me for years before he went to Novartis, and really missed Doug. And so, NCATS, Nate gave a background of it. So, we were founded in 2004. We were part of the Molecular Library Probe Center Network, LMLPCN. And really our role, as Nate said in the introduction, my role in particular was to really introduce pharma level high throughput screening and early chemical optimization. But the key piece, the longer I've gone in my career, what I recognize is we were a network. And so when I mentioned, we're talking about scientific collaboration networks, what are those and what are some of the fundamental principles? And I think, what I've learned over time and what has led to things like Aspire is the first collaborative platform NCATS really had, or even then it was called NCGC Chemical Genomics Center, was our big robotic system, which you see in the lower left here. Because what was this? This was a collaborative platform that would provide a resource that investigators could not have in their university because the complexity, they couldn't afford it, especially not part of a grant. And so, the first, I would say on my journey of setting up collaborative networks, this robotic platform was the first. And I think it's also important to note that these platforms can't exist in a vacuum, and they have to have part of a larger and a broader team. And again, this is part of this entire theme throughout this, presentation, is none of this is possible with a single individual. So when NCATS was first set up on the intramural side, there were 5 teams: adenine, thymine, guanine. Doug used to lead guanine, for the record, cytosine, uracil. And Nate, you actually were in guanine as well. So guanine seems to be where we get some outstanding people. Each team was comprised of biologists, chemists, and informaticists, and then every project was a collaboration with the respective external team. They brought the disease expertise, we brought the expertise with regards to assay optimization and screening. And our group that Nate mentioned, responsible for automation, compound management, we also have analytical chemistry. And so the goal was to really provide operation and maintenance to make sure that these platforms, and these are 3 primary platforms that we've had over the years. And again, the first 2005, it's still kicking. The system of 2005 is nothing like the system today. There's some components are alike, but these systems have all evolved. And I think this is another key piece of this. If you want to build a scientific collaborative network, you need to have things that are foundational. You have to have core components that you can build upon, and a foundation has to be exactly that. I expressed repeatedly in my career, I can't build on a foundation of sand. You need stability. And so the reason I'm highlighting these 3 systems, these are not the only 3 systems we have, but these were foundational to the MLPCN, what we were trying to do, and it led to a substantial amount of work, which again continues to this day. And so this was a example in my mind of a physical collaborative system that enabled science to the point Nate just said, where we've had, multiple INDs, we've had several aspects reach clinical trials, and it was all of this is the foundational support, really focused again towards assay biology. There is the downstream chemistry. From an automation perspective, by the numbers, just to give you guys some context, and these aren't entirely, these numbers are close, is what I would say. But from 2006 to now, we've run close to, between our Tox robot, a specific program, and our big robotic system, close to 400,000 1536 well plates, over 6 million robot moves, and those are supporting over 600 projects with close to over 5,000 individual runs. This is a huge amount of work. And I know, Nate, you and your team can appreciate this with the NCI 60. You guys are very similar in this, is where you have the foundational tools that you guys set up, and then you run multiple runs over the years to enable research. On top of that, from a compound management perspective, what have we achieved since we started this is, we've had over 1 million sample IDs created. FOTS is a system we have called, which is follow-up order tracking system, where anyone at NCATS and external can submit a request for plates, and they'll get quantitative high throughput screening style plates made, over 60,000, follow-up plates over 7,000, drug combination. We do quite a bit of matrix screens, close to 30,000 plates made. And all time, we've shipped over 5,000 compounds to 180 collaborators in 11 countries. And I think that's also important, that these collaborative networks are not just internal, they're not just national, they're international. And again, if you want to have these networks set up to perform complex research questions, you really have to have a foundation. You have to have components that you have to depend upon. And so really this led me, as I got further along in my career, and as Nate mentioned, I was sort of, I had a realization in around 2012, performing high content screening, that automation was becoming an IT problem, because the sheer volume of data we were generating, it used to be megabytes, it turned into terabytes, and now we're generating petabytes of data, was becoming so much that it was really becoming an IT problem. So I was given the opportunity to become the CIO of NCATS, and I took it really as a preemptive strike, because I wanted to make sure that IT worked for science and not the other way around. So this scientific collaboration network, I want to give an example. I've always loved this publication. This is actually old. This is from 2012. And what you're looking at here is, this is the 5-year data from Scopus and MAPS, each joint authorship of publications. You can look at these very complex networks. And once I became the CIO of NCATS, I realized NCATS actually has 2 of the largest networks in all of NIH. We have the Rare Disease Clinical Research Network, the RDCRN, which is over 20 active consortia, over 200 disorders, and it's a consortia of a consortia. It's over 200 sites across 4 continents with 3,500 users, over 40,000 participants. This is one network NCAT supports. Another network that NCAT supports is the CTSA program, 61 hubs, over 200 awards, over $600 million a year, close to $700 million a year now. If you look at NCAT's budget, this is by far the largest portion. As a matter of fact, this is the single largest grant program in all of NIH. And we have this network. And really, though, what we learned is that it wasn't a network at all, is these awardees are essentially silos. And then what we started seeing, we started getting data more and more that showed is that we identified institutional IT infrastructure as a cost constraint for the program where individual grantees were required to support their own capability. And there was really 3 downsides. One, we were using grantee funds for research for IT infrastructure. We don't want to do that. It really didn't offer any capability or incentive for collaborative efforts between and among individual grantee programs, which would require additional support. So if they built tools, they would build them for themselves. They wouldn't build them for the network. And then really the other one, and we had a real nightmare situation with this one, number 3, there was no easy way to transition work to NCATS when complete. And, you know, if you talk to a grantee and you say, what's your transition out plan? We've really gotten the answer in the past. What do you mean? What's a transition out plan? And so what we decided to do is we want to establish a common cloud environment for different grantee networks so we could purchase software and services wholesale. So total cost across the entire program is cheaper than the sum of their individual retail rates. So they get more and we pay less. And that's another lesson I've learned in my time working the government. The US government does something incredibly well is we buy things. So how can we leverage our buying power to actually benefit an entire network instead of an individual grant? And really, by doing so, what are we really trying to establish? We're trying to create a virtual research organization. So what is a VRO? A VRO is a manifestation of collaborative research networks and is cooperation supported by computer networks. What are the key elements of VRO is you have to have a shared research infrastructure, you have to have institutional identities and credentials. And this sounds incredibly dry, but without this problem being solved, you can't get anywhere. You need authentication. So if you guys ever hear authn, that's authentication. That means you are who you say you are, and then you have authorized, which is auth-z access, meaning you are authorized to see this. And then really, you have to have these identity and credentials span across organizations. And this is actually a huge problem. And this is, you know, an old graph or an old slide, but at this point, many of these problems still exist. And so the health research community needs to collaborate across the web with partners across organizational boundaries, sharing tools and data while maintaining security and confidentiality. And again, this is a very difficult thing to be collaborative and secure at the same time. It is far easier said than done. And again, these are real challenges. And so this was a pain point that NCATS was feeling. You know, if I want to provide and we want to set up a collaborative network, a cloud environment that could be used internationally, we would need something to manage identity and access. And some of this, again, is very dry, but if you want to enable world-class cutting research, you need to have foundational tools that you can count on. So we actually had to come up with a tool that we had several requirements. It needs to integrate with various source of identity, NIH login, Research University, social login, in common federation, has to integrate with cloud services, Salesforce, Google, Microsoft, has to integrate custom application, has to be easy to use, and it has to be convenient and secure access for the users. And we came up with something called UNA, and this is Unified NCATS Auth. It's free and open source for NIH. It's designed to support the NIH community. It's really small. It can provide multi-factor authentication, has multiple IDP support, that's identity provider. Again, with to really allow people access into the environment. And taking a step back, if we have a cloud environment, if I want to have people inside of it, I need to know who they are. So essentially, this is the front door. So we came up and we started working on UNA in 2016. This is when we first kicked off this project, and it went through an entire period of iteration to really start allowing researchers access into the NCATS environment. We had another project that kicked off, which was for FHIR. This is more on the electronic health record side. This was kicked off by a project with HHS and the ASPE program, where they wanted interoperability with different electronic health record types. we had to have a secure scientific collaborative platform. So just like we had the big robot to perform our screens, if I want to have a collaborative analytical environment in the cloud, I need a platform that's secure and that I can build upon. So we actually went out and working with NCI. Um, we were the first to pursue and start using a tool called Palantir. And as a matter of fact, we sponsored them getting their FedRAMP authorization. We also integrated this with Jira. And then a key piece, especially working with academic collaborators, is we had to adopt Google Workspace. And this seems minor, but at the time when I first went to NIH and I went to CIT and I said I want to use Google, they said, no way, we're Microsoft only. If you talk to many academic researchers and you say, hey, use Microsoft Teams or use Microsoft Word, they'll end the meeting with you because it doesn't work for them. And Google Workspace has been incredibly enabling. We went through this and we got an ATO for this. The reason I'm pointing this out is we want to build a large virtual environment. And then, as Nate mentioned, we got an award for this thing called N3C, the National COVID Cohort Collaborative. And the reason we could stand this up, so the birthday of N3C is May 4th of 2020 during the pandemic, is because we had already done the work beforehand. It didn't just spring from the ether. We wouldn't have been prepared if we didn't build everything before it was needed. And I just want to say that as a theme as we start thinking about Aspire and what's next. And so what is N3C? N3C is the largest publicly available free centralized linked electronic health record data set in US history, available for anyone on this call to go use right now. It represents all 50 states. We actually have capacity that we have identities or we have patient information of over 90% of the counties. So that's the map you're seeing on the left. This is the density population. Over 90% of the counties in the United States are represented within the N3C. It actually is very similar to the census data with regards to race, ethnicity, background. We have over 20 million patient level records. And then how do we measure success? And why am I talking about this virtual research platform that we've developed? We have over 5,000 users for 30 countries. We have over 90 studies. This has led to over 72 different grant awards, totaling over $100 million from almost, half of the NIH ICs. It has over close to 3,000 citations with an H index of 26. Again, this is 3 years old. And on top of this, this is a massive training tool for AI and machine learning. And I'm just sharing this as background to Aspire because we showed first for the MLPCN, we had a collaborative physical platform. For COVID, we set up a collaborative cloud-based platform. And the reality is for Aspire is we actually need a physical and a virtual collaborative environment at the same time. So this is no easy task, but it's an exciting work. So what is Aspire? Is reimagining the research laboratory ecosystem, and the acronym is a specialized platform for innovative research exploration. And you have a mixture of laboratory functions at the lab bench coupled with integrated automation solutions. And again, and this goes back to when we started, with the MLPCN and NCGC, we used to have what was called offline screens and online screens. So we do the assay optimization, assay development in an offline fashion in a traditional wet lab, and we would translate that assay to something we would run in automated fashion. Again, Nate, very similar to what you guys are doing with the NCI 60 of how you do your validation, then you bring it to an automated process. And the reality for Aspire is we need both. We need the modern lab bench where we standardize automation assisted template development. And then we need to translate those templates to amplifying them by bringing them to automated platforms to really get that power of scaling these up. And so really, what are we trying to focus on? And this is a publication that came out in 2019. And a lot of folks have really heard about this concept now, this design, make, and test cycle is we want to design molecules virtually. We want to make them physically. We want to test them, we want to analyze them, and we want to increase the iteration time. This is a very high level overview of this. And so what does this look like from the context of Aspire? What does this look like in the context of NCATS? Is we want to make sure that if you look at this chart, the green arrows in black are really pointing to inputs to the system. The blue are both outputs and data in. So we're going to have a biological target. We're going to optimize this assay. And with chemical libraries, we're going to run virtual physical screens. We're going to analyze that data. Based upon that data and the selection of compounds, we're going to perform chemistry route planning and do our synthetic analysis. And then we're going to create these compounds and we're going to automate chemistry. and we're going to feed those back into our chemical libraries and rinse, repeat. And so really from a perspective of Aspire, for years, we've done assay optimization at NCATS. We have chemical libraries. We've done virtual screening. We've done physical screening. We've done data analysis. Really, the pieces we're focused on are the chemistry route planning and the automated chemical synthesis. And this is where I want to take a second to have a pause, because what I've learned in my time, and we're all researchers and researchers have their quirks, biologists have their quirks. If I say I want to automate chemistry, there's chemists that pop out of the woodwork sometime. They feel like I'm insulting them somehow, which is very interesting, and I've never quite understood it. So I want to stress something. The better living through chemistry is when we say we want to automate chemistry, we are not saying that there's a deficiency in chemistry of any kind. As a matter of fact, when we talk to the chemists, they'll say, the chemists have changed the world, and they're right. So this is just an example. If you type into Chat GPT, what advances have been made in organic synthetic chemistry in the past 100 years? New synthetic methods, new reactions, new protecting groups, new materials. It really has changed society. The COVID pandemic could not have been a more fitting example of that. But just want to highlight that when we say we want to automate chemistry, we are not in any way diminishing the role of chemists. And I'm just putting that disclaimer out there because it's just been interesting to get some visceral reactions sometimes from chemists. If anything, we need more chemistry. If anything, we need more chemists. If anything, we need to augment the capabilities of the chemists because we need them to come up with the advances of the future. Now, the challenge is, how do we automate chemistry? And what are some of the bottlenecks and pain points in organic synthetic chemistry, making it difficult to introduce automation to the process? And really, this is a very complex and highly specialized field. And there's several bottlenecks and pain points that make it difficult to introduce automation to the process. And again, I'm going to share these slides. You don't have to read all of these, but complexity of reactions, lack of standardization, and really those 1 and 2 right there are some of the biggest bottlenecks that we've really been facing. And in particular, the lack of standardizations. Not only is there not standardization, especially when it comes to chemistry and automated equipment, there's no standards almost at any level, is we're all essentially talking another language. And then in many cases, we have to do tremendous energy to translate those languages with ontologies to come up with a common framework. And so those two alone are ones we've really been struggling with. But taking a step back is, if our goal is this very lofty, cloud-enabled lab of the future everyone has heard about, and we're going to want to focus on automate chemistry, where do we start? Is we want to start with automating chemistry. I mean, I need an indigo reactor. I need something in this case that can automate a chemical reaction in an industrial fashion. And that's a key point I want to stress, is if you look at the biological screening and the automation involved in biology, at this point, there's close to 30 years of development and billions of dollars of investment to make robust technologies. And I think about, the MLPCN, when we started this in 2005, is the difference in quality, between different pieces of equipment and vendors was stark. You had everything from this thing will never work to this is incredibly good. And that investment over time it's such, there's so much good automation out there. It's almost a question of, pick ones you like or what you can do with. So if we want to automate chemistry, we have to start with an indigo reactor. And this is us, again, a foundational piece. We had the big robot for the MLPCN. I had Una for a collaborative cloud environment. I need to have a rock solid foundation of a piece of instrumentation that I can say I can automate a chemical reaction. This can run 8 reactions in parallel. It allows for remote reaction setup and monitoring. It can go very low temperature, very high temperature, very high pressure, can actually stir. It's really a pretty incredible device. But the reality is, when we first purchased this in 2018, it was a prototype. It was nowhere close to being something anywhere ready that we could put in an automated fashion to run tens of thousands or hundreds of thousands of samples. And then on top of that, we have to come up with a common framework of reaction templates and then protocol representation. So what does that mean? How do we turn the chemical formulation into a program, essentially, that we can execute on the reactor to create the product that we want? And so what does this workflow look like? And this is, to go through this, and we'll show a video in a moment, is we're going to load the vial. And again, this can be either a human or a robot. We're going to put the vial into the reactor. We're going to load the recipe, purge the reactor, start the reaction, monitor it. In this case, we're using a PIN process where we have different chemical reagents within a vessel that essentially shatters either based over time or pressure. And then we're going to monitor our reaction, stop it, open the reactor, and analyze to see what we made. I'm going to show this little video, and this is not glamorous at all. This actually was a collaboration that we did out with a group called Stratios that was working with Eli Lilly. And this is the reaction vial, and you see the pin that he's putting in to the vial. He's putting that holder into the reactor, and then you're going to see this close because this is running at high temperature and high pressure. And I just want to give it a second just so you can see the device, because this is all parallel. It's sealing, and at this point, the reaction is starting. This doesn't seem glamorous, and you can see this coming down to actually make sure you seal that vial while the reaction is running. It took over 3 years of effort to reach this point that we could run that reaction. I also want to stress that if you think about assay screening, we say 1536 well plates. That means I have 32 by 48. A 96 well plate is 8 rows and 12 columns. So the current state of chemistry automation we have is a single column of a 96 well plate. So we have a lot of work to do, but we've shown that this can be done, and we can actually run these in parallel. So as one reaction is finished, we can actually put another reaction in there. And so I just want to show, in this case, an example. We were just doing a very basic amide coupling reaction here, where again, we would start the reaction, we would bring the pressure to 6 bar, the pin would break and that would release. We would heat the reaction to 60 degrees Celsius, and then would finish 3 hours later. We could bring that over to a mass spec, and we can validate that we made what we thought we could make. And so what this led to is that a key area NCATS is focused on is, how do we set up these reaction benchmarks of very common reactions to validate the performance of the reactor, not just at NCATS, but also at other facilities. That's another key piece of this is, we want to make sure that the data and the reactions running are reproducible. And then how do we continually improve this system for reliability and productivity? And then also adding additional automation capabilities. So we've added computer vision on top so we can actually see all the reaction vials. We've added stainless steel pressure blocks and pistons with chemical resistant coating. We've added rifling the adapter so it's easier to pick up to get it out of the robot. Again, these are the very little details that takes something from being a prototype to production, because our end goal is that this indigo reactor is essentially running close to 24/7. Because even though we only have eight reaction chambers, if we run these nonstop and we can introduce a new vial that we prep on a different system, we actually have a great chance of creating a substantial number of compounds. And so on top of that, a key piece of this as well is the software to control these instruments. And I want to highlight something which is a backbone of the Aspire project now, is if you don't have an API, we are not interested. We tell that to vendors. If there's no programmatic way to control your instrument, thank you for your time, no sale. And unfortunately, there's a lot of people in industry which have taken on, really a closed system approach, and it's just not beneficial, especially not for open science. And this is just showing some of the software improvements they've made. And also we can remotely control this with a mobile device now that we can log in and execute a reaction. So just want to give, again, a foundational piece is creating an automated reactor. But that being said, as part of the Aspire program, the reality is if we want to get the adoption of the chemists, we actually have to have core integrated benchtop chemistry capabilities that can work with the workflows they're already familiar with. And so we've really started, how do we develop new chemistry and with serial processing, and then to introduce this with a mixture of semi-automated tools that, if you look at this from a workflow perspective, is we have a high density storage solution we're building in-house. We actually bring that to our different reaction platforms. We perform a reaction. We'll also include the indigo in this. We bring this over for SPE chromatography. We bring this over to evaporation. We measure our product, we put that back into storage, and then with that storage, actually those compounds can make it to our compound management. Again, we don't have to build compound management, we already have it. We don't have to build analytical chemistry at NCATS, we already have it. So based upon those components we have, how do we augment those so we can go from development of new chemistry to production and scale up of different compounds to put to test. And all of this, I also have to stress, is being monitored and controlled with a dashboard, which is a very complex Internet of Things and IoT infrastructure that we're building from the ground up. What we're also going to start working on is how do we orchestrate all of these instruments. But a real key piece of this is to make sure we enable the chemists at the bench level. And we've had outstanding feedback and response from the chemists at NCAT so far. Now, it's not just the bench. We actually have to do all of this in concert with an enterprise level reaction analytics platform. This is the virtual piece. The chemists in the call know better than I do. Most chemistry these days starts virtually. They don't just get into a hood and start making a reactant. And so we've been developing what we call our AICP, the Integrated Computational Platform, where we have the reaction in our ELNs, we have our compound registration, we have our analytics, we have our biological data, and we're building a knowledge base where we have retrosynthesis, reaction prediction, condition prediction, reaction templating, hypothesis generating engine. Again, we need, what are the molecules, what are the targets we're trying to hit? What are the molecules that we should try to make? What's the most effective path to make them? How do we translate that chemical formula to a machine language template that can be put in a piece of instrument, and to create the vial, bring that vial to the reactor, run the recipe on the reactor? Again, very straightforward to say, an incredible number of components to pull off to do this. And so really what's the framework is that we're taking for the Aspire program is we want to digitize and automate the bench. And we have this NCATS dashboard with an IoT architecture. And then we also want to integrate that with an automated solution in our Aspire facility. So this is at NCATS for years. We've been in the same building. We're off the main campus. We're right across from Shady Grove Hospital. Right around the time of COVID, we were able to actually lease additional space and then design this. But also, again, I want to stress is Aspire requires both, and this is the importance of that virtual platform, is we're going to have virtual experimentation help guide what we should make and connect that to the physical ability to make it and analyze it. And that's a key step. I mean, a lot of folks these days have all this hype about AI and machine learning for drug development. And I talk to so many companies, they don't even have access to a wet lab. And I have no idea how they think they're going to be successful. Some of them are going to win the lottery, right? They're going to get lucky. They're going to find a molecule that's going to validate. But without that physical reality of being able to validate what you're making with regards to a biological target, good luck. And so what are we doing with our platform is we really want to build this automated closed loop processing where we're going to start with in silico design. We're going to do a preparation step, reaction to setup, control, monitoring, completion, isolation of our product for purification, and biological testing. The purification process itself is one we're focusing on, not just us, and is incredibly complex. This is one where the chemists that say it's very difficult to automate chemistry are not wrong, because there's so much complexity baked into the system that's very difficult to automate some of these processes. I did want to highlight as well that what we're also trying to do is build this in a module fashion, that we have modules that we can build to in a standard format. So it's not just essentially a free-for-all with no standardization, because, and people forget that sometimes too, especially when it comes to automation. The first step of automation is organization. And the automation problems and projects I've seen fail is where they have no inherent organization to their data, their research, their organization, and they expect to somehow automate something which is disorganized. It's really good luck to that. So I just have to stress, standardization and organization are critical to automation. So with regards to this space, we started construction during COVID, and unfortunately, there was a silver lining to this all happening during COVID, is everyone was working remotely, so we didn't have to worry about people where they were sitting. This is actually our new compound management space, so we had compound management and the Aspire space at the same time. And also, for the record, you folks are all relatively close. If you ever want to come visit NCATS, reach out to me. More than happy to give you a tour, because we work collaboratively with NCI very closely. Once it was complete, we had this really nice lab, and I think the lab stayed in this state where it looked relatively empty and free from stuff in about 3 hours. If NCATS does something well, it fills a vacuum of stuff. And the current state of what the laboratory looks like is this. And again, the portion over here, you see a glove box here and kind of in the corner. I know it's a little bit hard to see. The Aspire space right now is a little bit on the back end, because we actually had to have some lab space. That luckily this lab space came online, but then we were doing a project for a lot of COVID research, which was the antiviral prevention program, APP. So we work together as a team at NCAT. So we're sharing this space right now, but this equipment is ready to move to a different building. But then we're talking about the Aspire project. We're talking about this lofty work that we have to do. And if we, again, we want to go all the way from assay optimization to automated chemical synthesis and do this rinse repeat, is this project is too big for NCATS to do alone. And this is something, again, that I've learned in my time at NIH. And one of the things I'm truly grateful working at NCATS is, and a lot of NIH, most of NIH, there is a firewall between intramural and extramural. As a matter of fact, we've been thrown out of meetings where we were intramural and someone in extramural finds out, say, you can't be here. That doesn't exist at NCATS. And when I mentioned the project N3C, N3C was successful because NCATS acted as the foundation providing the collaborative cloud environment. But we could not have done it without the academic researchers, the hundreds of them that were leading this effort with us and who are responsible for its success. And so if we want to actually have Aspire succeed, we can't do this alone at NCATS. And this is where we take advantage of our extramural collaborators. And so we actually put out 2 RFAs. Is we wanted to have 2 hardware modules, meaning we wanted folks to focus on physical hardware that will end up in the NCATS laboratory, and then 3 virtual modules, which this will be working with groups collaboratively, where what they develop virtually will actually be part of the overall NCATS ecosystem. And again, taking a step back, this sounds straightforward. This is incredibly complex because I'm the CIO of NCATS, which means I'm responsible for the security and integrity of all NIH systems. And what we're doing in this case is we're saying we're going to deploy your tools into a federal environment and be the stewards of both your tools and your data. Most CIOs say no to that, and they're not wrong because we're assuming a great deal of risk. But without that risk, you cannot perform research. And that's another thing that sometimes at NIH just gets completely lost. We are not the National Institutes of Administration. We are not the National Institutes of Procurement. We're not the National Institutes of Security. We're the National Institutes of Health. And if we want to perform research for health, we have to take and accept risk. And I know that sounds obvious to many of the folks in this call, but I can tell you at NIH, that gets lost in the shuffle sometimes. And so we put these 2 different RFAs out, and we ended up getting 5 outstanding proposals and 5 outstanding teams. And so on the hardware side, we had the University of Glasgow and Dr. Lee Cronin, Purdue University and Dr. Graham Cooks. And then virtual side, we have MIT, Purdue, and CDD. And really what we were starting to build is a collaborative team. And so, and if anyone's familiar with the RFA process, you notice these first initiated in 2021, they weren't actually awarded until late 2022. And they're utilizing, in this case, UG3, UH3 mechanism, where essentially we give them funding to do work for the UG3 phase. And if they're successful with that funding, the UH3 phase, the next phase will be to bring their work, meaning their physical system, into the NCATS environment. The collaborative work has already started. So really what we did is we kicked off an Aspire Summit. And this was getting all the teams together 2 days, January 30th, 31st of this year. The next summit scheduled to be in Purdue, December of this year. And so all that being said, it started first with a site tour for them to come from NCATS, and we had an amazing response that we had all 5 groups show up and over 30 people. And then we had an entire meeting where we went through the different agenda and the different teams. The other thing I really want to highlight about this is before the summit kicked off, is all 5 teams signed a CDA where they agreed to share information and they agreed to work together. And again, I just have to share for the folks not from the extramural side, that is almost unheard of. Extramural funding and PI-driven research is incredibly competitive, and there's really, unfortunately, not much incentive to work together. There's not much incentive to work collaboratively. I think in my career, I've learned people love the concept of team science, but the execution of team science is really difficult. And so what are we doing? We have, what are these pieces that we're bringing? And so with the Cook's Lab, what they're really bringing to the table is reaction screening. And so they've developed a novel platform for high throughput synthesis and screening using a novel mass spec technique to synthesize and perform some bioassays on compounds at the nanogram scale. We're always about miniaturization. You lose less substance, you use less, you pay less. And then building an integrated hardware module to explore bioactivity with actual physical compounds. So this is a current, a make it system, as they call it. This was funded by DARPA. And really, this is based upon their DESI technology, this desorption electrospray ionization, where it's a technique that allows chemical information to be obtained directly from a wide range of surfaces And just using a 2D stage, DESI can be implemented with an imaging mode where the MS spectra are collected by rastering across the plate. And so in this case, the protocol is relatively straightforward, is they can really take a bioactive complex, they put that to functionalize reagents, they do a pin transfer, they spot that up to a 6144 sample plate, they put that in the DESI, we get our spectra and we analyze. And this can be used for quite a bit of different techniques. And what else is really exciting about this now is they're building the next generation of this with Hamilton and Waters. And this is something that NCATS has been actively involved with them, because the goal is once we build this DESI platform for the Hamilton as the liquid handler, the Waters as the mass spec, is they are building this in Purdue, really as we speak. They're about to go through the site acceptance test. Is actually at the factory for Hamilton right now, but this is going to be shipped ultimately over to NCATS. And so what's exciting is this hardware module that they're developing extramurally is going to become an integrated component of the NCATS Aspire platform that researchers can use. And again, hopefully globally, very similar to what we did with the MLPCN. And just the concept of that, if you go to certain program offices and you say, hey, an extramural funded program is going to have equipment in an intramural funded lab, you should bring your fainting couch for them because they'll pass out. They won't understand the concept. And so really, this is just an example of some of the technology that they can perform here. And this is just a late stage formulation of a complex molecule and showing with this technique, not only can they do reaction screening, they can actually run chemical reactions as well. So these technologies, we're trying to get things that are versatile. And another thing I really want to stress too is NCATS specifically focused on getting extramural awardees that augmented our capabilities, not that duplicated them. Because again, this is a very complex, broad program. There's no value in us duplicating or having people work in a parallel track. The next program, the next awardee was also from Purdue. There's quite a bit of good research going on Purdue, and this is a collaboration called Links and Links of what they call, this is the 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 ledger for interactive query and execution. And so essentially, this is a blockchain-based approach that will essentially allow data access automatically and securely in a distributed fashion on blockchain. And it's also going to be able to distribute and schedule work in a common laboratory environment. This is a really interesting one because really what they're doing is they're coming up with what they call a laboratory coordinating utility. And this utility is software that can bridge between computational, so the virtual design, and execution on a physical instrument, and to do this in a distributed fashion on a blockchain-based approach. And I want to stress this too, and this gets to a research perspective, is some of these are very exploratory. Some of our research, it doesn't mean we're going to have adoption, but we're going to try. We're going to experiment. And so we've been working with them already, that we've set up, we've given them an instrument that's running an LCU, and we're going to set up in the initial run parameters. We're going to run the instrument. We're going to analyze the results, and then we're going to feed that back, and they're going to design these molecules and the different formulations that will all then be displayed and saved in the database, and where we can visualize and schedule this. We've started really small on this, or we started basic to get started. We literally just gave them a dispenser and we said, we want you to install the LCU on the dispenser that can coordinate with your blockchain. And we want to be able to read and write from that blockchain so we can initiate and execute an experiment. There's another key piece about this is the inherent security and confidentiality associated with blockchain. So this is a really interesting one. And I have to say a big part of this too is the research schedule they're coming up with in a very basic fashion. How can we pair system and data parameters in a conjunctive normal form, and then do an evaluation check, with basic statements. So if we have a system and we have data, so our system type is assay with 6 plate capacity and 80 wells per plate, and we were looking for a purity of high or medium, and then we have requested assay, is how can we actually schedule a process to turn that into a notional command set, evaluate those commands to see, hey, can this run or not? And if it's true, to actually execute that. So this is, again, very high level and a really interesting one, I have to say, conceptually, because I've done a lot of things in the automation space. I would have never considered having an instrument be controlled via blockchain. So it's a really interesting approach. We're excited about this one. We've already done quite a bit of work deploying the LCUs in multiple instruments and having them essentially send commands from Purdue and Indiana and control instruments in Maryland. So it's a really challenging, and this is a fun one. The next one was collaborative drug discovery. This is actually an ELN. Many of you may be familiar with this. And really what they want to do was take virtual approaches to new chemistries and really orient questions around how to provide rich contextual molecular representation, and then also create the representations and annotation of molecules that have questions of bioactivity, question of chemical reactivity, and then to really utilize that well annotated data. And this is just examples, some of the screenshots of their application. If we use that to help generate deep learning models to help us design novel molecules. And so really a key piece of this, everyone hears AI, if you don't have proper annotations and data annotations for your reactions of both your chemistry and your biology, designing machine learning models around that incomplete data is just setting you up for failure, or you'll get lucky, one or the other. And so this has been a really interesting collaboration so far, not just using their tool, the commercial off the shelf, but the research component to actually come up with these different machine learning models based upon the data and the annotation. And so really our goal with this one is to continuously build models, but to have integration points with APIs at multiple levels, from the property of the data upload to the interactively explore, to generate the suggestions, to build the models. And the one thing I'll give CDD credit for right now is they have amazing support for us, and we actually have our collaborative cloud environment. This is another point I should have made earlier. Our collaborative cloud environment is not a single tool. It is a collection of federated tools that have to have connectivity via APIs or messaging. And so what's been really awesome about working with CDD so far is we have their tools fully integrated via API with our cloud environment utilizing our single sign-on. Again, it sounds very basic, but it's incredibly complex for it to be seamless to the user. The last thing we want a user is, if we send down a user and say, hey, log into 20 different systems and you do the work to make them connect, they're going to vote with their feet. They're not going to use it. So that seamless integration is just absolutely critical. The fourth group, MIT, they were really building virtual informatics modules. This is Dr. Connor Cooley. And again, their whole focus is on how do we have informatics and machine learning modules for reaction planning, scheduling, simulation, and optimization in the Aspire Autonomous Laboratory. Key piece of theirs, they're really focusing on all of these, but a lot of it is also in the simulation. How can we actually run simulations virtually before we do anything physically? And I think that's becoming more and more prevalent in the space of research because, again, we can run things virtually far easier than we can run things physically. Running things physically takes a lot of work. Like I like to tell people, we've run, 500,000 plates, you know, since 2006 and 1536. And that took a Herculean effort. That's a huge amount of data. The first computer process they can do, 0.5 megahertz was made in the 70s, to give you perspective, right? So we think it's a huge number of operations. Computationally, that's nothing. And so really in this case, this is oriented around computer-aided synthesis. Given a target molecule, can we use algorithms to develop a feasible synthetic plan with inexpensive commercially available materials? So can we come up with a target? Can we come up with a plan to make it? Can we use cheap off-the-shelf components? And can we make that molecule? And then really, can we do this in an iterative fashion that we go from a goal definition, we design it, we synthesize it, we validate it, we model it, and again, do this in an iterative fashion. What else is really cool is they were also part of the Make It program, and really is to enable this goal of a self-driving laboratory through algorithmic decision-making and information-centric discovery. I do want to say one thing that was really interesting about this proposal as well, and this concept, is we had to be very specific to the reviewers, and this is where the extramural performance and the scientific review. Scientific reviewers are hard coded and hardwired to look for what's novel. And that's great. But sometimes we don't want what's novel because then you reinvent the wheel. And so we had to explain to the reviewers, we want you to evaluate things that already exist. And since they had put a huge amount of work into a tool called ASCOS, which has already been developed and is already deployed, is we didn't want them to be dinged in the review that they were proposing to use a tool they already invested in. And I know that sounds crazy to a lot of us in the intramural side, but in the extramural side, in some cases, this focus on what's always novel, it can become a detriment to actually making things that are functional and stable. And I think that's just the nature of the world, right? Everyone, if anyone goes and requests budget and they say, hey, I need more budget to create this new thing, you have a far higher chance of getting it. Then you say, hey, I need more budget for operations and maintenance. That's where oh, I don't have any money for that. But without that operation and maintenance of existing tools, you're not getting far. And so this is where they've come up with interactive path planning, how we can come up with those reaction paths, create the proper selection, turn that into something that we can actually visualize the results. And then based upon that, how do we schedule that? How do we simulate that? But ultimately, how do we bring this to something that we can actually make? And I think, again, this is really a key piece of this. Ultimately, we want to start virtually, but the real power of the Aspire platform is how can we turn this into things that we can actually make? Because we want to make molecules, we want to test molecules, and then based upon those results, we want to make additional molecules. And this is where it's a huge team effort. And so finally, the final piece or the final winner was for a physical module was Dr. Lee Cronin's lab in the University of Glasgow. With what they have is two real core components. Is they have the computer. Is so the computer is a really it's a physical device that they developed to put into a hood to run chemical reactions, and that's really important. So the computer is a nice piece because it's a physical reaction vessel that can create molecules. But what's even in some cases, in my mind, even more important, they developed a universal digital chemical standard they call CHIDL. And so really, they say that the computer is a chemical executable based upon Python code embedded in this KDL language. And really what this means is that you can turn chemical reactions into code that then you can execute on different pieces of hardware. And that's a key piece, not just the computer. And one of the projects we're working on now is can we use KDL and can we use their structured programming language to actually run instruments that are not necessarily just theirs. And so again, a lot of this collaboration comes down to how to automate chemistry, and then automation follows from digitization of chemistry, convert written prose procedures and protocols into machine readable formats. And again, in this case, ubiquitous and agnostic machine readable formats, not a machine readable format that I can read on a Beckman, or I need to create a new one to read on, a Hamilton, a universal machine readable format, and convert the ecosystem of labyrinth instruments into digital representations with defined physical information interfaces. And again, this is so important because then you can model this virtually to come up with things you can make physically. And so again, automation closely linked with success in simulation optimization. We want to run as much optimization and simulation before we do anything physically. And so really, this is giving an example of slides they prepared of the current state of all the steps that are involved, which is taken to actually get the result to your purity. And what they want to do digitize is turn all of those current steps, which are analog, and turn all of those into digital representations that can be simulated, but they can also be executed. And this has been really exciting. And so this is where they're talking about the different reactions to robotics, is how you have your reaction abstraction from reaction workup, isolation, purification, turn that into a description language, bring those to hardware specific commands, and actually bring that to a modular robotic platform and what they're calling the computer. And this is just the background of their KDL blueprints, as they call these. Again, this is just for the folks familiar with programming. This isn't anything fancy. This is essentially XML, JSON, YAML. This is no different. How are we coming up with this to build these blueprints in this XDL file, this KDL file, and how can that file be used to drive our automation? So this is just showing the working prototype that they have, where they have the modular hardware, they have their chemical programs, they have a working prototype. And one of the things that's really exciting about this project and what we're doing with the collaboration is they have their grant to develop this in Scotland. But as we're working with them right now, we're currently building a KDL in our own environment. And so again, this computing is code to molecules. How do we turn code into a molecule that we can analyze and test? Really exciting. And the different modules that they're coming up with is really cool. They're coming with photochem, redox, different types. Is they're introducing different modules to the computer. This one on the lower right is very similar to the one we're doing right now, which is really basically bioreactor. We're starting with a very basic approach, but again, we're building a computer and running it with KDL and NCATS as we speak. And then the goal is that we're going to initiate a chemical experiment from Scotland to run at computers in Glasgow and computers in Rockville. And we're going to compare the results at the same time. And so again, this is just showing the modular nature. So we had this really exciting workshop, and really the outcome was for the collaborators to come and to have a much better understanding of NCATS, our priorities and capabilities. There's really no substitute for in-person engagement and team building. And the goal ultimately was to articulate key roles for each collaborator that we saw them as filling. And I think this was very important for them because, this is a very broad project. There's a lot of different players, but I think it's important for the grantees to understand what is it that we want you to do. But again, the way we look at this at NCATS is the end state is there are none of these barriers, right? There, there's puzzle pieces we use as a representation to show, okay, this is the clearly defined work. But the reality is we want to work as a cohesive team between the extramural grantees, the NCATS intramural program to automate chemistry, to drive discovery forward. So I appreciate you guys listening to me drone on for the better part of an hour. And it's a huge team effort. I just have to share this massive team effort. This is the NCAT side. The core Spire team is everyone here. And I just really want to highlight Alex Godfrey, who is a consultant, but he's really, the consultant lead of the program. Worked closely for years with Carlene, Chris. We could not do the extramural work without Dota and Sean Gardner, the program officers. Our scientific director, Anton, again, just a massive team. And obviously this team is not complete without the extramural collaborators as well. And it's not just the PIs, it's actually their entire labs. And I think that's what else has been exciting. We interact with the different labs. And this also gets back to a key point that I learned early in my career at NCATS on the extramural side, is we have access to some of the smartest people on the planet. Why don't we work with them instead of just giving them money and not learning what they're doing? How about we work together? And so with that, I think I'm right at time, or I don't know if I went over, Nate, but I think we may have a minute left for questions.
Sam, that was incredible. Yes, I would love to open the floor for questions. You touched on so many different things. Please, colleagues, does anyone have a question? If not, I'll start, Sam. So I'm really curious about, this is a really nice overview of kind of the foundation and movement towards this final Aspire platform. And I'm sure that this Aspire is going to continue to evolve, right?
Yep.
So, but I'm sure you have kind of sort of like a minimum viable production in mind. What's the approximate timeline for when, the entire working system will be, kind of?
It's a great question. And there's great questions in the chat, by the way, and I'll respond to these. And sorry, I wasn't looking at the chat when I was presenting, so I may follow up with folks. There's great questions, but I would say it's a great question, Nate, and the minimally viable product that we're shooting for is we identify a target, have potential compounds of interest, get reaction pathways of what we think to create those, perform reaction screening to find the most optimal routines of the optimal conditions for those reactions, create the reaction vial that we can then run on the indigo reactor, perform the reaction, bring the reactant to our analytical chemistry, and see what we made with all the components in a manual fashion. And I think we're going to be there by the end of this year. And I'm super excited about that because I know that doesn't sound like, again, like some of the chemists are going to say, I do that every day. It's like, but to do it in a potential that can be automated. That's what I have to stress. All of those steps I just mentioned, we're going to do the equivalent of a sneaker screen, but to have it that we were showing that we have the capability to bring that closed loop and then say, okay, how do we set the stage to run this in an autonomous fashion? The one question Jim asked in the chat, it's an amazing question about the DESI, qualitative or quantitative. I don't want to say it's a loaded question, Jim, but different people will give you a different answer. I would say, I think, the peaks don't lie. So I'd say it's quantitative, but there's aspects which are qualitative. But yeah, so the MVP to us, Nate, though, is showing the components are working together, even if they're not fully automated, because that shows the possibility of once they are connected, that we can have this run again, and ideally that autonomous fashion, if that makes sense.
Yeah.
And then I will say there's another good question from Dwight about are there lessons learned from Stratios Lilly? And absolutely, it's, and sorry, I know I'm cherry picking, but I think it's a great question from Dwight. And and we learned many lessons. I think some of the lessons we're learning, don't try to do too much. And this also gets back to, one, really focus on what we want to do. We went into Aspire with this lofty thing. We want to create this translational laboratory, and we still do. But that being said, we really want to focus on what we see as a gap, which is the lack of standardization and um, automation capabilities for chemistry. And that alone is a huge thing. And I think one of the lessons we learned from Stratios, what they were trying to do is I think they were trying to do too much. And that's, I'm not knocking them. Again, like the Stratios, that project was incredible with their ambition, but sometimes it's a classic case. You try to bite off more you can chew, you try to boil the ocean, it can become overwhelming. So one of the real lessons it really helped us focus on is what are the core capabilities? And I think that really highlighted our need to make sure we work with the chemists, that we bring value to them at the bench today to put ourselves in a position that they'll adopt automation tomorrow. So I don't know if that helps answer your question, Dwight, but I definitely think there's lessons we took from that. But what really was focus our energies to where we think we can make impact.
So Doug also asked, our in vitro ADME assay is going to be part of the SCR and Aspire workflows? This is a really interesting question.
They will be. It's a great question. I didn't show. That's why I'd love to bring anyone for a tour. Is one of the things we didn't highlight, at NCATS, is we have automation, we have compound management, but now since about 2015, we've been doing a ton of automated tissue culture, and we also have full DPMK and ADME capabilities. And part of what we also want to work on are going to be standardized assays that we can run against these compounds. So the answer is yes. But again, for full disclosure, those are going to start essentially in a semi-automated fashion. We're not going to have that fully integrated with the system. We'll get compounds of interest and we'll bring that to our ADME group, and say, hey, these are the compounds, and then we want you to run these against the battery of assays that you guys already have very well characterized. So it's a great question. So the answer is yes, but I again want to give the caveat. I don't want to say, oh yeah, we have everything fully integrated. We're nowhere close to that yet, but we have the components and we can get the data.
Does anyone on this call have any questions that they want to ask Sam before we close?
And I'll definitely share these slides. And again, anyone is always welcome to come visit NCATS. And part of this presentation as well, I just want to stress, like, we are working with folks at NCI on this already, so it's, I really look at us, it's a very collaborative effort.
Yeah, it was incredible. I definitely want to see it. If there are no other questions, with those of us that are here today, I want to thank Sam for that fantastic presentation. It's an incredible amount of work you're doing, and it's so exciting. I mean, this is really, pushing the boundaries of what's possible with, screening and probe and drug development, and enabling the, extramural, public sector. So that was the other question I wanted to ask, Sam.
Yeah.
In the long run, when this is completed, it's like a functional Aspire platform, which will, as we discussed, I'm sure continue to evolve. What's the vision for how the community will access this?
Yeah, no, it's a great question.
Would it be like a grant mechanism, or maybe it's not known yet?
It's a great question. I would say it'd be, very similar to what NCATS has done, even during the MLPC. And I do think, our hope is that there could be grants to utilize this as a resource. Because that is important, to have the extramural collaborate, the community. We do need to fund those researchers. I will say that's something, I will say I didn't truly appreciate until I, got exposure to the extramural community, the intramural community, for all of, the challenges we face. We at least have stability on the extramural side, and the PI side is very challenging. So, part of this as well, the reason we agreed to work with intramural extramurals will always be an extramural component. So right now the extramural component is towards develop and build the platform. And I'm hoping once the platform is ready to be used, then we can actually have the extramural component actually start issuing grants to utilize it, right? That's the goal. And I think that's exciting, because again, build this platform, have the capabilities, but also the amazing chemists that are in there in the extramural space, not just chemists, biologists, researchers, to make sure they have funding opportunities to actually utilize the platforms.
And it seems to me that people might use, just depending on their needs, maybe just certain aspects of the Aspire platform, right?
That's right.
Like maybe I'm a chemist and I would really like the system to synthesize a compound and send it to me, right? But I don't necessarily need the testing of it in
That's right.
biological assays. And people would be conceivably able to do those kinds of things, just use parts of the Aspire platform.
It does, actually. And I will say that's also what's going to be interesting as part of this UG3, UH3 mechanism we have right now, is part of the UH3 will be for them to say what a research question will be. So we're sort of doing a pilot. That's what's interesting. We're building this platform. We're also doing pilots on what it could look like for folks to utilize it. And maybe they don't need the whole thing. Maybe they just want one component.
Mm-hmm.
And so, and again, this is one of those, we won't necessarily know until we try it. You know what I mean? And that's, again, this gets back to the part of taking risks. That's the part that does drive me crazy sometimes is, folks sometimes, tell me the answer before you run the experiment. Well, how? I mean, it's like, I got to run the experiment first, then I'll get back to you. So sometimes there's an expectation. Well, oh, this is risky. We need to know everything before we start. Like, it's easier to move the car when it's driving. You know what I mean? Try to
Yeah,
steer in place. It's really hard. Once you're moving, you can steer. And so I do think we're going to find some of these things out.
absolutely. Does anyone have anything that they'd like to bring up before we end it? Okay. Thanks again, Sam. That
Yeah,
was
thank you.
incredible.
Thank you
Thanks,
guys for having me.
thanks, colleagues, for joining.
Bye, everyone.
Goodbye.